We're in Chapter 10, and this video lesson will cover sections 10.3 through 10.6. Like I said in your previous video lesson, we will skip around a little bit at the end of the chapter to save a little bit of time. In 10.1 and 10.2, if you haven't watched those yet, make sure you go back and watch that video lesson. In this video lesson, we're going to go into the two reactions that take place in photosynthesis into greater detail. So make sure you watch that one first. In 10.3, it says the light reactions will convert solar energy to chemical energy. So we're going to take that sunlight and turn it into sugars. And we're also, in the process of doing that, going to make some ATP and some NADPH. Remember, NAD is our electron carrier that is going to pick up those hydrogens when the water molecules get split apart. And so the chloroplasts are kind of like little solar-powered chemical factories. Thylakoids is where that light reaction will take place and then turn that chemical energy from the sunlight into ATP and NADPH. And then eventually get passed along to build C6, H12, O6 in the second part of photosynthesis, which is the Calvin cycle. Okay? All right, so it says the nature of sunlight. Light is electromagnetic energy. It's also called electromagnetic radiation. Electromagnetic energy travels in rhythmic waves. We call those our measurement for that is the wavelength. It's the measure of the distance between the crest of electromagnetic waves. It can range from less than a nanometer to more than a kilometer. And so that's how we will measure our energy that is coming from the sun. And we calculate that or put that onto what we call the electromagnetic spectrum. That's the entire range of the electromagnetic energy or radiation that's coming from the sun. And we call that our visible light. And so the visible light drives photosynthesis and produces the colors seen by the human eye. And so here it is, a little picture of it or a chart. And you may remember this little saying, Roy G. Biv. That's supposed to be an R. Roy G. Biv, which stands for red, orange, yellow, green, blue, indigo, and violet, which are the colors we see here, red starting here and violet ending over here. Those are the colors of the rainbow. And so all of those colors are visible in the white light that is coming from the sun. And so certain wavelengths of light are going to be used in photosynthesis. It says light also behaves as though it consists of discrete little tiny particles called photons. And so those photons are little bits of energy. So it has a fixed quantity of energy that is related to the wavelength of that light. And so those photons of light are what the plant will pull in to the chloroplast. And so pigments are substances that absorb visible light. Make sure you know what pigments are. We're going to talk about a very specific one called chlorophyll. And chlorophyll will absorb violet, blue, and red light. While it is reflecting, so it's going to reflect back to our eyes that green light. So that's why plants look green when we look at them. If you had on a red shirt, your red shirt would have red pigments in it, so all of the other colors of the rainbow would be absorbed into your shirt, and the red would not. It would be reflected back to our eyes, and so your shirt would appear red. It says we have three types of pigments in chloroplast. The main one will be chlorophyll A, and so that's going to capture the light for us for photosynthesis. We also have chlorophyll B. It's an accessory pigment. It's also used, but not as much as A. And then there's a couple of other accessory pigments called carotenoids. These are the ones that are yellow and orange, and They will absorb light at different colors, usually at violet and blue-green. And so this allows photosynthesis to take place on a larger spectrum. It says some are also photoprotective, that is, they absorb excessive light that would otherwise damage chlorophyll or react with oxygen. And so some of these other accessory pigments are also used in photosynthesis to kind of keep the reaction going there and keep from damaging the chlorophyll that's there. It says the absorption spectrum of chlorophyll A indicates that blue and red light will work best for photosynthesis, and green is the least effective. Because the plants will reflect the green and not absorb that light, then photosynthesis would not occur. It says the action spectrum for photosynthesis is a profile of the relative effectiveness of different wavelengths. It confirms the effectiveness of violet, blue, and red. So those are the two main pigments. Those reds and blues get pulled in for photosynthesis. Green gets reflected, so it is not good for photosynthesis. We had this man, Theodore Engelmann, back in 1883. He was the one who first demonstrated that there were different wavelengths of light. He performed a couple of experiments, and he showed that over here in the blue colors, chlorophyll A and chlorophyll B were able to perform photosynthesis. We see here in the middle of the spectrum there that the green wavelengths of light, no photosynthesis was occurring, or very little anyway. And then we see that photosynthesis started to occur again here in the red and orange section there of the spectrum. And so the rate of photosynthesis here in the middle declined when those pigments are exposed to the green wavelengths of light. It says in the last decade, so here recently, we've also discovered some other forms of chlorophyll, chlorophyll D and chlorophyll F, and they can absorb light at even higher wavelengths. And so certain bacteria, cyanobacterium, are able to do this. The chlorophyll by light excitation. So we're going to see that these photons or bits of light are going to be pulled into the chloroplast, and we're going to see that it's going to cause electrons to become excited. It says when a pigment molecule absorbs light, one of its electrons will go from the ground state to an excited state. That allows it to become unstable, and when it is unstable, we're going to see that it's going to very easily react. And so here in our picture down at the bottom, we see our photons, which are our bits of light energy coming into the chlorophyll molecule. That will cause electrons that are here in the chlorophyll molecule to be released and enter that excited state. And then they will be available for reactions to take place. And we're going to talk about a couple of reactions that are going to take place inside of the chloroplast. And so where these reactions will take place, we call that a reaction center complex, and we call them photosystems. And so we're going to have a couple of photosystems that we're going to talk about, and it's going to be made up of a reaction center complex and then a light harvesting complex. And so don't get all caught up in this part of it. I want you to understand more of what's taking place there. We're going to name our photosystems, but don't really worry about what they are made up of, okay? But the light harvesting complexes will transfer the energy of the photons, so that's the energy of our light that's coming in, to a chlorophyll A molecule that will be found in the reaction center complex. So that's the main idea of what's happening here. We're going to take the energy from the sun, transfer it over to the chlorophyll molecules that are found at the reaction center complex. And so here's a picture to help us understand what's taking place here. And so it says a primary electron acceptor in the reaction center accepts excited electrons and is reduced as a result. So here's our sunlight coming in, our photons, bits of solar energy. This little purple molecule here that's our complex. So it's made up of a light harvesting portion and then the reaction center. Here's the reaction center up at the top of it, okay? And so when the sunlight comes in, chlorophyll A, here's the chlorophyll A molecules down at the bottom, right here. Chlorophyll A will accept those bits of energy, and it will cause electrons from those chlorophyll A molecules to move up to the reaction center complex. Okay, and this is going to make more sense here in just a minute, because we're going to talk about some other molecules coming in here and where those electrons are going to be coming from. Okay, and so there are two types of photosystems in the thylakoid membrane. So remember, when we are talking about these photosystems, we are talking about the light reaction that takes place inside of the thylakoid membrane. So that's where we are. And we're gonna, these are named kind of backwards a little bit because of when they were discovered. So photosystem 2 is also called the P680 because it absorbs light at the wavelength of 680. We have photosystem 1, which is called P700 because it absorbs wavelengths of 700 nanometers, okay? And so this is what, we're going to use this diagram to help us understand what's going on in these two photosystems. So energy will enter, light energy will enter into photosystem 2, then it will move over to photosystem 1. Okay, so we're going to do 2 first and then 1. And they were discovered at different times, and so that's why they're almost backwards, okay? So we're going into 2 first and then 1. Okay, so, and still remember, we are in the thylakoid. Remember, the thylakoids are those little round discs that are green because they contain chlorophyll. And so within the thylakoid membrane, light energy, so number 1 will take place. We're going to go through a series of 8 steps that are going to take place inside of these photosystems. And so the light reaction we were talking about earlier, we said that light will come in, water will come in, water will be split apart, oxygen will be released, and the hydrogens from the water molecule will be picked up by NADPH, and we're going to make some ATP in the process. Okay, so that's the simplified version. We're going to go through what's really happening here, and there's 8 steps, okay? And so those 8 steps, we're going to break it down. Number 1, so here's number 1. Our light comes in to photosystem 2, and the energy is going to be passed among the pigment molecules until it excites P680. So right here, P680, that is our chlorophyll A that is accepting some light energy, and it's going to cause this electron to move up to the primary electron acceptor, okay? And so now P680 is turned into P680 plus. Okay, and so far light has come in, it's caused that electron at the P680 photosystem to move up to the primary electron acceptor. All right, next, number 3 happens. In number 3, here we are, right here in number 3, we're going to split a water molecule into 2 electrons and 2 hydrogens, okay? And so the hydrogens are the proton portion of the H, and we've got an oxygen in there also, okay? So it's going to pull the electrons. So we're going to get 2 electrons. We're going to get 2 hydrogen protons. If we pull the electrons off, the only thing that would be left were 2 hydrogens and 1 oxygen atom, okay? When we do that, the hydrogen, here's the hydrogen right here, will be released into the thylakoid space. The oxygen will kind of hang around for a bit and then combine with an oxygen from another water molecule that gets split later to form oxygen that will be released. And so here is the oxygen that is leaving from the plant, will eventually leave out of the stomata, okay? So in this portion right here, in step 3, here's the water molecule. The water molecule comes in to the P680 photosystem, photosystem 2. It is split apart. Those electrons, here's the electrons that came from it, get picked up here or attracted to the chlorophyll A. The hydrogens get pumped out into the thylakoid and the oxygen is also released, okay? And that's step 3, okay? All right, in step 4, these electrons that were originally found on the hydrogens from the water molecule being split get passed along a series of proteins here in the electron transport chain. We had an electron transport chain that we talked about during cellular respiration, but the electron transport chain has a series of proteins. Here they are, there's their names. I don't care that you individually name them, but just know that as those electrons are passed along that electron transport chain, we're going to make ATP while we are pumping out hydrogen protons out into the thylakoid space. And so the energy released by electron transfer pumps those hydrogens into the thylakoid space. And once we get a bunch of hydrogens out in the thylakoid space, we're going to create something called a proton gradient. And that proton gradient, once you get a bunch of them out there, eventually, due to having a gradient, they're eventually going to have to cross back across that membrane. And when they do that, they're going to make some ATP, okay? And so that ATP is going to be used a little bit later in our Calvin, what we call the Calvin cycle, to make some sugar molecules, okay? All right, so that's step 4. Let's move on to step 5. In step 5, so here's our step 5, and we just talked about it a little bit. The potential energy that was stored in the proton gradient, remember you get a proton gradient because we're pumping all of those hydrogens that were originally part of the water molecule. Remember, we stripped the electrons from them, and so now they are protons. All they have is a little positive proton on them. So it says in photosystem 1, so here we are in photosystem 1 on this side, the transferred light energy will excite P700, that's our primary, it's going to excite those electrons up to the primary electron acceptor again, okay, in photosystem 1. And then they are going to be passed along but this light will cause those electrons to move up to the primary electron acceptor, so that in number 7, in our 7th step, those electrons will pass down this electron transport chain again. So this is a second one. This time we're not going to create a proton gradient, but instead those electrons right here that were originally on the hydrogens that were split from the water molecule are turned over to NAD to make NADP+, okay? So remember in our original diagram that we were looking at in the previous video, we said that we were going to create some ATP and some NAD plus, NADPH, from the light reaction. And so that's where this is coming from, okay? We made some ATP right here. We made some NADP plus right here, okay? Using photosystem 2 and photosystem 1 to pull the electrons off of the hydrogens from this original water molecule that came into the thylakoid of the plant cell. Okay, and we'll go back over that again in a minute, but let's finish up with step 8. So in step 8, NADP plus is going to collect those hydrogens and become NADPH. Those electrons were originally found on the water molecule, and so that's where they came from, okay? It says the formation of NADPH also removes that H plus from the stroma, okay? And so here's our two products that we have made, NADPH and ATP from the two photosystems. Okay, so let's go back to our original diagram. And so everything we just talked about in those 8 steps are happening right here in the light reactions in the membrane of these thylakoids. Okay, the water molecule comes in, sunlight comes in, oxygen is given off, and we made these two products, ATP and NADPH, in photosystem 1, or photosystem 2, I guess, and photosystem 1, okay? And so those 8 steps occur so that we can make these two products to start the Calvin cycle, okay? And remember, all of those products we just made came from using light to split a water molecule. This is why plants have to have water, and they have to have sunlight, okay? All right, so here we go again. I'm gonna go over it one more time. Water molecule comes in, the electrons are pulled from the hydrogens, they enter into the photosystem 2 P680 electron acceptor. They get excited from the light, okay? At the same time, the oxygen is released from the plant. It's no longer needed. Those electrons move down the electron transport chain. In the process, they're pumping these hydrogens out into the thylakoid space and creating ATP. Okay, that's where the ATP comes from that we're going to need in the Calvin cycle. All right, those electrons move down the electron transport chain and excite the electrons at photosystem 1, P700, and those electrons move up to the primary electron acceptor. They move down the electron transport chain again. This time they're not creating a proton gradient, but instead these two electrons that were originally found in the water molecule way over here, okay, get added to or picked up by NAD to form NADPH, okay? And so there's our two products from the light reaction that were made from that original water molecule. Remember, oxygen is given off as a waste product in the process of doing that. And for all of this to happen, you have to have the light energy from the sun, okay? And remember, right here, these little pigment molecules are called chlorophyll. So this first reaction and this second reaction, P680 and P700, would not happen without those chlorophyll A molecules being there. Okay? We can also refer to these light reactions. Sometimes you might see them referred to as a little mechanical analogy going on there, where the photons get added to the photosystem. It excites the electron. Those electrons move down, creating ATP. Photosystem 1 gets some more light energy added. electrons get excited, move down another electron transport chain to make that NADPH, okay? And so that's just a little picture you might see sometimes showing what's going on there inside of photosystem 2 and photosystem 1. It says the chloroplast and the mitochondria both generate ATP, so just remember that we are making ATP in both of those so that these processes can be fueled that take place there. We're going to kind of move on for time here. It says, although the spatial organization of chemiosmosis differs slightly, there are similarities. Just know that both of them are making ATP that is used to fuel these processes. So in the mitochondria, they are pumped out into that intermembrane space. In the chloroplast, they're pumped out into the thylakoid space. And that proton gradient is what creates that ATP that is made in those processes. It says both ATP and NADPH are produced out in the stroma side of the thylakoid membrane, making them, so these two products will be used in sugar synthesis over in the Calvin cycle. So here they are, both of our products will be sent out here in the stroma for the Calvin cycle so that we can build sugar. Okay, this is just a little diagram here kind of relating it all together. So right here, here's our chloroplast that are used here in the photosystems, okay? And so these photosystems, I'm going to clear my ink here so we can kind of get started. This diagram here is showing the thylakoid membrane. So this is the membrane of a thylakoid. Sunlight comes in, water comes in, the photosystem 2 reactions take place, electrons get moved, water is split, oxygen leaves, and the hydrogens get pumped out into the stroma, okay? And we have that same enzyme called ATP synthase that does that for us. In the process, we're making ATP. So that's the first part that takes place. Then those electrons get excited from photosystem 2, and they get moved along a series of proteins called the electron transport chain over to photosystem 1, where more light energy enters into the photosystem because chlorophyll pulls it there, chlorophyll A. Those electrons become excited again, creating NADPH in the process. And both of those two products, ATP is sent to the Calvin cycle and NADPH is sent to the Calvin cycle. And there's going to be one more thing that will enter into the Calvin cycle. We know that plants consume CO2 from the atmosphere. So we're going to see that CO2 will also enter into the Calvin cycle so that the plant can build its sugars. Okay, so that's the main idea for the Calvin cycle taking place. This is an animation of what's going on. I recommend you watch that. All right, we're going to move on. Section 10.4. We're going to break down the Calvin cycle into its individual steps also. But remember the overall idea, it says the Calvin cycle, we're going to use the chemical energy from ATP and NADPH. Remember those two things were made in the light reaction, and we're going to pull in some CO2 and build sugar, okay? And so plants will be able to make their own food. It says carbon enters the cycle as CO2, so plants are taking that out of the atmosphere and pulling it into their leaves. Remember, plant leaves have those little tiny openings called the stomata, and they're going to make a sugar that we're going to call G3P. Okay, G3P can be used to make glucose later. Okay, and it says for net synthesis of one G3P, so in order to make one of these molecules, we must complete the cycle 3 times. Don't get all caught up into these numbers, but we're going to understand the main idea of what's going on here. And so if it does that 3 times, it's going to pull 3 molecules of carbon dioxide out of the atmosphere in order for this to take place. Okay? And so in phase 1, this is where the CO2 that is coming in from the atmosphere is going to bond with a 5-carbon sugar named Rubisco. Okay? So just remember Rubisco has 5 carbons. That's supposed to be a 5. I think I made it worse. And so if we combine these two together, carbon dioxide has 1 carbon and Rubisco has 5, we're going to create a 6-carbon molecule. And so in the first phase, let's go in our picture up here. We're going to use the diagram as we move along. So we are in phase 1 right now, which is called carbon fixation. And so remember we just said in order for this whole cycle to take place and to make 6 of these G3P molecules, we're going to have to take in 3 carbon dioxides from the atmosphere. But we're going to look at it as if we just take in 1 carbon dioxide and make the cycle happen with just 1 so that we can understand what's going on per carbon dioxide molecule. Okay, so for 1 CO2 molecule to come in, it will join up with Rubisco, which is already there. It's a product that the plant recycles, okay? Remember, Rubisco has 5 carbons. We've got 5 carbons, and remember, CO2 has 1 carbon. When those combine, we're going to create this 6-carbon molecule right here, okay? In the process of making that 6-carbon molecule, we're going to break it down through this cycle. And when we break that molecule down, we're going to have some ATP to get that started. And we're also going to use those electrons from NADPH that were made. Guess where both of these 2 products, or these 2 reactants that we need, came from? They came from the light reaction. So in order for this Calvin cycle to take place, we've got to have ATP and NADPH made from the light reaction. Okay? So this 6-carbon molecule right here that we created, or that the plant created, will then flow through this cycle and be broken down. Okay? And so as it gets broken down, we're going to call this next phase the reduction phase. So each molecule will be altered by what we call phosphorylation into 6 ATP and reduced by 6 NADP to produce this G3P sugar, okay? And so NADPH and ATP will be used to break this 6-carbon molecule into 2 of these G3P molecules, okay? So that's our main things that we're starting with here. So for every CO2 that comes in, we're going to make 6 of these molecules. There's 6 of these right here, okay? I said 2, but I meant 6. All right. Now, when 6 of those molecules get broken down or get sent through the cycle, 5 of them are going to keep going through the cycle, and only 1 is going to come out and be used to make glucose and other organic compounds. So only 1 will be considered a net gain, okay? And then we're going to regenerate Rubisco again. So after this G3P is produced, the other carbons that were left over from these 6-carbon molecules will then come back up and become 5-carbon Rubisco again after they are phosphorylated. And so this will require another input of ATP so that Rubisco can be recycled and reused again. Okay, so we've got to have ATP again for that to take place. Where did we get that ATP? That ATP came from the light reaction, okay? I don't care that you know all of these names of everything that's taking place here, but just know that CO2 has to come in to the Calvin cycle, which is taking place in the stroma. We are creating these G3P molecules, which is our sugar, or used to make sugar for the plant cell, okay? And so here's our overall diagram again. So now we're on this side. We've got CO2 coming into the Calvin cycle. Remember, the Calvin cycle is taking place in the stroma. So this is happening in the stroma. ATP that was made in the light reaction and NADPH that was made in the light reaction enter into the Calvin cycle. That series of steps takes place where Rubisco joins up with carbon dioxide, forms a 6-carbon molecule that gets broken down into G3P, which is used to make our sugar for the plant, okay? Rubisco then gets recycled, brought back up here so that it can pull in more carbon dioxide, okay? Here it is again, and the diagram here is a little more complicated, but Calvin cycle happens in the stroma, carbon dioxide enters, builds a 6-carbon molecule, ATP is used, NADP is used from the light reactions to make these G3P molecules, which get recycled to make Rubisco again to keep the reaction going. And then some of them are used by the plant to make glucose and other organic molecules to feed themselves. Okay, so that's just a quick overview there of what's taking place. This is an animation of the Calvin cycle. Here's another one. Make sure you go watch those. It says for the net synthesis of one G3P molecule, the Calvin cycle will consume 9 ATPs and 6 NADPHs. And remember, these two molecules, NADPH and ATP, are made in the light reaction so that these G3P molecules can be made in the Calvin cycle, okay? It says the light reactions generate the ATP and NADPH, and then G3P is the starting molecule for making glucose, sucrose, and other carbohydrates, okay? In 10.5, we're just going to kind of talk about a little overview of that one. We're not going to go into that section a whole lot. It says alternative methods of carbon fixation have evolved in hot, arid climates. Dehydration is a major challenge for terrestrial life for plants. And so in arid climates where they don't have a lot of water, remember you got to have water for the light reaction to take place, plants have made adaptations to help conserve water and come up with other ways to perform their photosynthesis. And so if you want to go through section 10.5, you can read about CAM plants and C4 plants if you want to, but we're going to kind of skip that section there in 10.5. All right, 10.6 is just an overview. It says photosynthesis is essential for life on Earth. And so here's your review. It says the energy entering the chloroplast, so that's where it's all going to start, is going to come in as sunlight, and it's going to get stored as chemical energy in organic compounds. The sugar will be made in the chloroplast. It's going to supply chemical energy and carbon skeletons to synthesize the organic molecules of cells. And so we're going to use these for energy, and we're going to also use it to build other organic molecules inside your cells. It says plants will store excess sugar in the form of starch in chloroplast and other structures. They'll use it to build their roots. They may store it in tubers or for seeds or for fruits. And so photosynthesis is essential for life on Earth. Okay, so there is photosynthesis in chapter 10. Okay, we're done.